Uh, in basically, the microscopy can broadly be considered as two different entities. One is called a transmitted light microscopy, other one is called reflected light microscopy. This is based on the applications that we use it for different observations. In addition to this one, the microscope can also be divided based on the anatomy. What kind of microscope? If the microscope is used to see a slide, then I say a compound microscope, because the microscope will have a lot of lenses, mirrors to form an image. So it has got multiple compounds of lenses that can be used to enhance the resolution as well as the magnification. So microscope is basically used not only to magnify but also to resolve the subject. So what is that we resolve? So as you, the name itself says that like microscope can resolve at the micro level. That means if something which we cannot see it in millimeter, that we normally observe it under naked eye, can now be resolved better, which are in the size of microns. Now the microscope has even gone to the level of where you can even resolve at nanometer level. So, and then now we can even if you can even gone to the era of called super resolution microscope. So other one is called stereo microscope, like where if you want to see the large organisms, for example, if you want to observe an insect,
Dr.
if you want
Ganesh,
to,
sorry to interrupt. Are you sharing your slides
yeah, I'm just
with
giving you a
participants?
brief idea, giving you
Okay,
a
sir.
brief
Okay, okay.
idea.
Then fine, sir. Then fine.
Okay. And then afterwards I'll switch over to, because I am just directly, I don't want to get into the advances in microscopy. Just I thought like I'll just explain you basics of this one. So stereo microscope are basically used, it's also called as a dissection microscope, where you put your larger specimen and you observe it in the form of a stereo, like where you will be able to get the three-dimensional effect. Coming to the compound microscope, it can again be brought it into two divisions. Like one is called inverted microscope, where you will be using it to observe cell cultures where cells are being grown, where you have to observe from the bottom. The objective is placed at the bottom surface, and on top of the objective you have a stage, and on top of the stage you will have a specimen which might probably be in the dish. Whereas the upright microscope is the one like where you put a slide and the objective is on the top of the slide, and then you will be observing. As far as transmitted light microscope is considered, the light comes from the bottom surface, it passes through the slide, and then it passes through the objective, and then it reaches your eyepiece. So these are some of the major components that we see it in the microscope. One should be the light source. It should be properly illuminated, and to make sure that the light is properly illuminated, we use a device called condenser. Condenser is the one like which collects the light from the light source. In earlier days, like it used to be a mirror where we used to keep our microscope in front of a window, and then we collect the light and we direct the light towards the condenser using a mirror. But nowadays, if it is electronic light, so you have LED light sources, you have halogen lamp, the light is switched on, the light is directed towards the condenser with a mirror, and now the condenser collects the light and it condenses, it makes a uniform illumination, and it makes to fall it onto a specimen. Where do we keep the specimen? The specimen is kept on the stage, and that's why we call it a substage condenser, because the condenser is just below the stage. Stage is the place where we place the slide or any other material, and we clamp it with a clip and then make sure that it is immobile, and then we allow the light to pass through the specimen. When the light passes through the specimen, if it is unstained, you may not be able to image it. That means it should be properly stained to the specific region of interest which we want to see. So there are different kinds of dyes are available. It can be hematoxylin, it can be eosin, it can be uh, aceto or different dyes are available that has an affinity to go and bind to a specific region of your tissue, and thereby making it glow with different colors. It can be lipid or it can be different kinds of. So once you illuminate the light onto the specimen, so the light will pass through, a phenomena called diffraction occurs, and then some light will pass through the specimen, some light will diffract, some light will be reflecting. What we are interested is the one which diffracts. When it passes, since you have dyes, these dyes has got an ability to get absorbed. The light will pass on to the specimen, the light gets absorbed. Suppose if you have a pink light, only the pink light is passed through, the rest of them are absorbed. That's how you see different colors. Now this diffracted light is collected by the objective. Objective is considered as the heart of the microscope. Objective will further resolves it, magnifies it, finally it delivers the image into another object called eyepiece. what does the eyepiece do? Eyepiece will collect this information, magnified image. So there are different kinds of objectives are available based on the magnification. We call it as 10x objective. It has an ability to magnify 10 times, 20, 40, 60, or 100. Different magnifications are there based on your magnification requirement, based on your resolution requirement. You choose to select a specific objective, and it will be on the optical path, and you will be able to observe those specimens. Once the objective forms an image, the image will be now delivered to your eyepiece. Eyepiece will have further additional magnification. It can be 10 times or 15 times. The total magnification is the magnification of objective into the magnification of your eyepiece. Suppose if you have a 100x objective and you have a objective of 10x, the total magnification what your eye receive is 1000 times. That's how you form an image. So that's the anatomy of a microscope. Nowadays, this is the digital era. So you don't want to see only, but you want to digitize it, you want to record it. So the microscope will allow the light to deliver the image onto a dedicated camera. Now we call it as a trinocular microscope. So normally we'll have a monocular microscope where you have one eyepiece. Binocular is the one like which comes with two different eyepieces with which you will be able to see through your eyes. And the trinocular is the one where it will have another eyepiece where it will deliver the light to a camera, and the camera is capturing the image, and it can directly be delivered onto your laptop or your PC. So that's how you form an image. So this is called transmitted light microscope. As I told you, the light comes from the halogen lamp. It is delivered to the condenser. Condenser will condense the light, and then it makes a uniform illumination and falls it onto the specimen. The specimen is stained. The light passes through the specimen, and a phenomena called diffraction occurs. And after the diffraction, the light is collected. The diffracted light is collected by the objective. Based on the magnification of the objective, the image will now get magnified and resolved. Finally, it gets delivered to your eyepiece. From the eyepiece, it will fall onto our retina where the images are formed. If you want to form an image on your camera, instead of making it to fall on your retina, you will make it to fall onto your camera chip where the information is collected. It gets digitized and the image is formed onto your monitor, where you can collect the image, you can freeze the image, and you can use it for all your printing purpose. That's how an image is formed. So any doubt here? Okay. So assuming that so the basic light path you understood, I'll let you know what is reflected light microscope. So now in this case, suppose if you have an opaque specimen where light will not pass through the specimen. For example, you have some metals, you have some materials which will not allow the light to pass through. You have some chitinous subject. You might probably have some wing structure where the light is not passing through the specimen, but you want to observe. You want to have a metal piece where you want to see the structure. You have to have some fossil structures, or you have some plant material where the light is not passing through. In all these cases, a transmitted light microscope may not be able to give you a proper image because the light is not passing through the specimen. In such cases, you need to have a light coming from the top. It will get directed towards the specimen through the objective. And now this is called reflected light microscope, where the light comes from the top. It allows the light onto the specimen. It allows the light to shine onto the opaque specimen, and the structures which are being shined will get reflected back to the objective when it is collected. That is why it is called as reflected light microscope. So basically, we have two different kind of microscopy: transmitted light microscope and reflected microscope. In the transmitted light microscope, we further divide it based on the contrast technique. If you have stained, then we call it as a bright field microscope, where you will have a bright background. For example, you stain it with hematoxylin, eosin, acetoarsine, GIMSA, or in G banding, R banding of your chromosomes. In all these cases, the light will pass through and there is a stain. And with this stain, you will see the stained specimen with color, and unstained specimen gives you a complete bright background. That is why we call it as a bright field microscopy. But this is not the solution for all your imaging. Sometimes you might probably want to observe zooplanktons or phytoplanktons. You probably prompted to collect a small drop of water from your pond, and you put it on a slide, put a small cover glass on top of it, and you observe none of them are stained unless and until there is some chlorophyll is seen. Then you want to see it. That means now in this case, you don't have the luxury of staining them, because if you want to stain them, you have to fix them, because you don't want to fix it because you want to see it in live. In such a cases, The transmitted light microscopy, which we were using for a bright field imaging, may not work. Now, instead of this one, we allow the light to pass through in oblique fashion, and we call it as dark field microscope. I'll just give you an example. Suppose if you are in a dark room, and if you switch on all the tube lights, you will be able to you will be able to see the people who are there inside. They are stained, but you will never be able to feel there is a dust particle. You switch off all the light in your room, the room become completely dark. Slightly open the window, then the sunlight is peeping inside the room. Then you will be seeing the dust particles from the ray of light which is coming inside. The dust particles are seen only when there is a complete dark background, a small amount of light peeping inside the room with the dust particles which are floating on this one. What happens now? Because of this oblique illumination, now light will not pass through this opaque specimen. It will reflect. The reflected light is collected. This is called as dark field microscope, where you introduce a cone of light into the specimen, and this cone of light will allow you to reflect the specimen from an opaque floating specimen, and that's how you generate a wonderful image of the floating zooplanktons or phytoplanktons in a droplet of water placed on a slide and a cover glass on top of it. Thus, we call it a dark field microscope. Now, coming to the live sample, where you might probably culturing some cells. It may be live cells or a fixed cells. It may be plant cells or an animal cell where you want to see, but you don't want to stay in them. That means, again, the cells are transparent in nature. You want to observe a technique called phase contrast microscope, where we will introduce the light with different phases. And if you see the sample which are there inside the cell, which is a cytoplasm, has always a higher refractive index. It is more viscous, more dense than the outskirts, that is the surrounding media. Now you have a nucleus. Nucleus has a nuclear membrane, and you have nucleoplasm, and it is more viscous, more dense than the cytoplasm. You have cell organelles. Again, the cell organelles share different thickness, different viscosity. That means when you allow the light to pass through this specimen, the light is penetrating with different speed because they are being retarded. Because of this retardation, because of this stopping of the light will move with different speed and reaches the detector or your eyepiece. So because of this change in speed, light passing through the nucleus or through the cell organelle or through the plasma membrane, so you can actually generate a shade, and this shade is now called as phase contrast microscopy. Phase contrast microscopy is basically used to see unstained thin specimens. Suppose if you have a thick tissue, like if you have a Drosophila embryo, if you have a mouse tissue, if you have a mouse brain tissue, in all these cases they are thicker. Then in such cases, there is another technique called differential interference contrast, or DIC, which will help us to see the unstained specimen whose thickness is more than 10 micron. Okay, so in all these cases, we are using microscopy with different kinds of transmitted light microscope. Suppose if you want to observe different cell organelles together, then you need somebody has to unmute. I think, can you please mute?
Why don't you show microscope, sir?
I'm just coming back to the microscope now. I just want
to understand.
Yeah, just a minute.
Show the microscope means we can see at least one time.
Yeah, just a minute. I was just coming to that one. Okay, now this is the topic. Can you see this one?
Yes, sir.
Okay. So now, so this is how a microscope looks. This is an inverted fluorescence microscope. What does a fluorescence do? Fluorescence is, you must have heard in your +2 classes, it's a phenomena of a chemical substance which gets excited when you allow a specific wavelength to pass through, and it absorbs some amount of wavelength and it emits. This is called fluorescence. What we do is we use this as a tag that can go and bind to different region of interest of your cells or the tissues. The ability to go and bind to different tissues, a different region of interest, or different organelles entirely depends on the chemical property of these fluorochromes. The name itself says fluorochrome means it has an ability to fluoresce. Chrome means color. How does it do? So these fluorochromes has an electron and proton and neutron. So the electrons will be rotating or in an orbit. When you give certain amount of energy, the electron will jump to higher energy level and it will not stay there for a longer duration. It will try to come back. When it tries to come back, it loses certain amount of energy, but it will give you some amount of energy back, and this energy which is coming back when it comes back to the ground state is called fluorescence. For example, if you are exciting a fluorochrome called DAPI, if you are exciting with blue, the excitation happens when the emission comes, it comes from the green. Similarly, if you excite with the green, it gives you a red emission. If you excite with red, it gives you. So this property has been used to stain different cells or cell organelles. In this particular case, we have labeled this is a dividing cell. This is the nucleus where you can see they are in the anaphase. So this dye called DAPI has an ability to go and bind to histones. So that's why the moment you allow this dye to go into the cell, it will specifically go and bind to nuclei, and your nuclei will start looking blue. Similarly, other dye called Alexa 488 labeled with a specific antibody, and this has got an ability to go and bind to the proteins of cytoskeleton. In this case, this is an actin filaments. It will go and bind actin filaments. Wherever you have actin filaments, it will start glowing. So the third color is actually a dye which has an ability to go and bind to the outer membrane of the mitochondria. The dye name is MitoTracker Red, and when I use this one, it will go and bind to mitochondria. So the result is now I could label, I could probe 3 different organelles within the cell, and I can observe and I can see whether there is any interaction between these two or not. If it is labeled on a live specimen, I can practically track them what is happening when the cells are in live condition. So this is what called as fluorescence microscopy. So how do you label it? Suppose if you have a primary antibody, this antibody has an ability to go and bind to a specific protein which is inside your cell. Now I have a secondary antibody. The secondary antibody can go and bind to the epitopes of the primary antibody, but the secondary antibody is tagged with a fluorochrome called FITC or Alexa 488, and this fluorochrome will now be shining only on those regions where your particular protein is there. In this case, if you see an image, the red color is a mitochondria. The dotted structures are the protein. So the red mitochondria specific dye will go and bind only to the mitochondria. The dotted structures are the protein that will go and bind only to those regions where this particular protein is there. In this particular case, this particular protein is localized on the mitochondrial membrane. That's how you will be able to image. This is a fixed specimen. Then suppose if I want to image a live specimen, what you do? So you will go back to the central dogma of molecular biology, where a gene is there. A gene produces a mRNA, and this mRNA is translated into a protein, and this protein will have different biological functions. Probably, like I assume that like a specific protein is responsible from cancers. Then I want to identify how does this protein behaves when it is there in the cancer, and if I treat them with a specific drug, what happens to this protein, whether the concentration of this protein will reduce or increase, or will it stop producing. So these are some of the questions that might probably come across when you are doing a live cell imaging. So in order to do this, you probably want to know at what time they are synthesized, in which location of the cell they are synthesized. Are they interacting with other protein to give a different physiological processes? In all these cases, what you have to do is you have to first label a protein which can give you a fluorescence color. So this protein is called now as green fluorescence protein, or GFP, and I tag this green fluorescence protein DNA along with the protein of my interest DNA, and now they are co-synthesized. What happens? Let's see here. Now this is a protein called DNA for green fluorescence protein. Now I have a DNA for actin. I combine them, and then now I will allow it to express. Then wherever my actin protein is synthesized, my green fluorescence protein also gets synthesized. So that means it will shine only on those places where my actin filaments are there. Then how do you observe it? Now I need a microscope which has an ability to excite a fluorochrome and then collect information. Those microscopes are called as fluorescence microscope. So in this case, I have a mercury arc lamp which has an ability to excite different fluorochromes. And now I allow the light to pass through a small filter called excitation filter. This excitation filter will block all other wavelength except for the wavelength of the light that is required to excite your fluorochrome, in this case your GFP. It will excite, and the GFP gives you a green emission, and the green emission is collected by your microscope. So all these things is done by a small box called a filter cube. Similarly, there are different filter cubes for different colors. And finally, when you observe it, what happens? You do is see a small spot, but this small spot, because of a phenomenon called diffraction, so it will give you a point spread function. Because of this point spread function, your image will never look sharp, it will be hazy. So in order to make it sharper, so there are different techniques. One such technique is called deconvolution. So this is an image where you collect a blur image. When a software processes it, it makes it sharper. So what you do is, now you have a thick tissue. Now I have acquired a fluorescence image, but now I want to optically section it, because the tissue cannot be physically sectioned at a thinner. So the microscope has an ability to optically section at a slice thickness of 10 nanometer. How do you do it? So these are some of the images, like where you see here, this is a nucleus, this is cytoskeleton, and a protein which is binding onto the nucleus. So you practically want to optically section them instead of physically sectioning them. A microscope called laser scanning confocal microscope was introduced. How does this do? So instead of using a mercury arc lamp, we use a laser, and this laser will penetrate into the tissue, and it shines a specific region. The image information is collected back through a small aperture called pinhole, and it blocks all the light which is coming above the focal plane and below the focal plane. Whenever you have an objective, the objective will always focus the specimen to a specific region called focal plane. And that means anything above the focal plane and below the focal plane are called out of focus light. The light which is coming from above and below are blur. But when you place a small aperture called confocal aperture or a pinhole, this pinhole will allow light coming only from one particular plane to pass through. The light which is coming from above is blocked. Light which is coming from below is blocked, and you get a sharper image, and this image is now collected. So this is an example. This is a pollen grain which is seen without the presence of a confocal pinhole. You see the entire thing, but the same thing when you use a pinhole, you can optically section and collect the information in the form of a nice section. So your sample is physically intact, but you have optically sectioned. There is no physical damage, but you are collecting the information from different planes. Then how do you do it? Like, so how do you collect an optical section? What we do is, so we collect a region here, and let me just go to this particular plane. See here, this is the first plane. I collect the information, I register it on your monitor. Then I go one step up. The previous step is out of focus. This plane is in focus. It is collected. Now the next plane, third plane is collected. Like this, from top to bottom, you collect the information one by switching the focus position from lowest position to the highest position. And now you have this information. This is an example of a Drosophila compound eye. We collected the information at different focal plane. How does it look? If you click here, so you see they are serially collected information from top to bottom. Once you have this information, your software can construct a 3D. This is called three-dimensional imaging. So you have optically sectioning. You have optically sectioned by rejecting the out of focus light. Each and every plane was sharp enough, and now you have a three-dimensional image. Once you have this one, you can generate a volumetric information. You can practically calculate the length, the depth, and this one. Now, once you have this information, can I take a cross section of this? So you have acquired the image from top to bottom. Now you have an apple, and can you cut this apple side by side to see the distribution pattern of your seeds? Or you have taken a tomato and you have sliced it from top to bottom, and now with this information, I want to see the distribution pattern of your seeds. So the software allows you to take, this is a soft knife. Now you are taking orthogonal slice view, with which you should be able to see the distribution pattern of a particular protein. This is a zebrafish eye. The information was collected from top to bottom, and now you constructed a 3D. And after constructing the 3D, you are slicing it, and you can rotate it. That's the beauty of optical sectioning and fluorescence microscopy, as well as confocal microscope. So this is how you do. You collect information serially from top to bottom. You pile it one above the other by the software, and software allows you to give a 3D projection as well as the optical slicing. This is an example of the neurons penetrating at different tissues. So you can see it is serially collected from topmost layer to the bottommost layer. Every layer you get some information, but you will never be able to measure the length and breadth of your neuron. But if you construct a 3D, you should be able to rotate, and you should be able to clearly measure, and you should be able to clearly see the morphological features of the neuron in its 3 dimension, for which you never had an access unless and until you physically cut it. Okay. Now the question is, I'm seeing fluorescence, but how do I see that they are coming from the cell? That means you also need to get a transmitted light image. The transmitted light image is the one which I was telling you. It can either be a dark field image, a phase contrast image, or even a difference image, a differential contrast, where it will give you the whole image of an unstained specimen. In this case, I'm getting a fluorescence from the reflected. The transmitted light passes through, and now it is collected. So in this case, you see here, this is the image. What you are seeing in the green is actually a particular protein which is around the nuclear membrane. The image which you are seeing in the gray color are the complete cell boundary. So this helps us to acquire an image at different direction. This is an image of the Drosophila head brain. So in this, the one which is there in the gray color is the transmitted light image, unstained image, the whole brain. The one which is coming from the center is actually the fluorescence signal that is coming from the central part of your. So this is your optical lobe, and this is the brain region where the fluorescence protein are synthesized, and they are localized in the brain region. So how do we collect? The confocal microscope will have different detectors, and these detectors will help us to collect different wavelengths. What do we do with this one? Okay, let's see how do we collect this one. So now I am exciting with particular color. I put a small detector in front of this one. I set the bandwidth to a specific wavelength, that is the emission bandwidth, and collect it. Okay, now let's see what happens. Just let me just come back to this one. If there are two different colors, which are one is a nucleus, other one is the microtubule, if there is an overlapping, we can clearly separate them using a confocal microscope. Okay, coming to the other image. So here there is a tomato root tip here. The tomato being a plant, it is known to give you a lot of autofluorescence, but this autofluorescence will share a different wavelength compared to the fluorescence of the GFP. So in this case, if I just say, you can see two different positions here. One is a complete green background, which is your autofluorescence. Other one is a GFP. A technique called spectral unmixing or spectral fingerprint help us to separate the autofluorescence and then the GFP, which gives us an idea that from which part of the tomato root tip the GFP are being synthesized. Other advantage of confocal microscope is called high dynamic range imaging. So in this particular picture, you might probably have seen there are two images. One is an image which is taken in the dark background. Other one is with a higher exposure time. So in the first image, you can see very good image of the places where it is properly illuminated, but you will not be able to see the buildings at the background. But if you increase the exposure time of your camera, you will be able to collect more light. The brighter region has become more brighter. The darker regions which was not visible earlier is now visible. This is called high dynamic range imaging. This is just an example. So this is $1 million night scene of a Hong Kong city. So now, so as a biologist, I say I'm okay with this image, but probably I would like to count the number of boats that are floating here, but the information that are coming from the boats is very less. But if I try to collect more light in this one, this might probably become, see here. Now you will be able to see the number of boats, but these regions become brighter, and some of the brighter regions become saturated. This can even happen in your plant cell or animal cells. So in this case, this is a plant tissue section. Now I am seeing some small structures here. In this case, the intensity is very low here, but these regions are brightly fluorescing. I'm fine with this one, but my idea is how much of signal is coming from these regions, which is in the core. Probably it is a xylem or a phloem. But in order to collect this information, I have to increase the exposure time or increase the incident light intensity. Now you can see a clear structure of these regions, but these regions are saturated. So in order to get this one can sequentially collect information at different exposure time. Now you will be able to form an image where the brightest regions are clearly seen without compromising the structures of the lighter region. This is called high dynamic range imaging. Nowadays you must have seen HDR or HDD, high dynamic range imaging in your camera, high dynamic range imaging in your mobile, or HD monitors can be seen. All these HD monitors, the one which has an ability to give you the brightest signal without compromising the lowest signal. It gives you brighter. We call it as brilliant information. Now the question is, can we perform live tissue imaging? Yes, it can be done. So the confocal allows you to image live tissue as well live cell imaging. So in this case, you have a heart of a zebrafish. You see here, the entire live zebrafish can be kept under the microscope, and now you are imaging with fluorescence as well as the differential interference contrast to see the platelets which are moving inside. So this entire image was acquired with 30 frames per second using a 512 pixels in X direction and 512 by X direction. But now what you say is like, I'm fine, but like I want to see the movement of the blood. So I want to have a better speed. Normally conventional confocal microscope gives you around 8 to 10 frames per second. But the latest advantage in confocal microscopy is to go for a high speed scanner. See, that gives you 100 frames per second. Now you can actually see the blood is coming inside the heart and then it is being pumped. That's how you can image in the confocal microscope. Not only that, so now in this case here, you have an image like where you see here, blood corpuscles are passing through this retinal artery. Now I want to see how does it move. See here, you can actually track them one after that. They are moving in one direction. See here. So this is called live tissue imaging. Suppose if you are growing the cells on a dish and I want to image, this can be useful for people who are working on drug delivery experiments. So like you might have identified some novel drug, or you might have probably extracted some plant material which you feel that it has got a anti-cancerous property, or it has got some like anti-diabetic property, where you are trying to see. Now I culture the cell in a small dish, keeping them inside the CO2 incubator. The cells are healthily living inside. Then I label them with respective colors. Now this is a microscope stage where you have a multi-well plate. You are growing cells on each and every well. Now if I image them, see here, each of them are being imaged. You can image for 24 hours because this entire sample or the dish can be placed in a small incubator, and this incubator is placed on the microscope itself. Now you have actually generated a laboratory on top of your microscope. Now you are imaging, you can see the cells are dividing. At any point of time, it allows you to add a drop of drug and then see the effect of a drop of drug of a known concentration onto these cells. So that's how you can perform live cell imaging. In this particular case, multi-point time-lapse imaging is done on HeLa cells for 17 hours of observation. Now, in this case, see here, this is a fantastic example. This is called NK cells. These are natural killer cells. So what you can do is, if you want to treat, nowadays the latest technology is like identify the cancer cells, load them with some antibody that can be identified by the natural killer cells. Now, in this case, the rounded ones are the cancer cells, and the green ones are the natural killer cells. Now, when I allow the natural killers to invade, initially the cancer cells, when they are in healthy condition, it will have a blue boundary, and this blue boundary is a cue for the natural killers to identify these are the cells that I need to go and attack. If you see the description, say natural killer cells mediated cell killing after therapeutic antibody application. The antibody is the blue one, which will go and bind onto the cancer cells. It will go and bind only onto the cancer cells. GFP labeled NK cells, this is green fluorescence labeled cancer natural killer cells, will now go and attack. If I start this once, initially most of them are in blue. Okay, then once this green cell will start attacking, the moment they die, it will turn into red. So over, see now this is 24 hours of imaging. So 30 minutes of imaging. So you can actually track and when actually the natural killer cells will go and attack the cancer cells and then kill them. So for to do this one, you need to focus the specimen in a same region, and it is done by a device called drift compensator. It will make sure that your sample is in focus throughout your experiment, and it will allow you to image your samples hours together. Suppose if you want to do drug delivery, you keep on imaging, you add a drop of drug. The moment you add a drop of drug, your sample may go out of focus. So in this particular case, you are trying to do an experiment called FRET. FRET is an experiment where you try to understand the interaction between two proteins. If these two proteins are together, then one protein can transfer the energy to the other protein. The one which transfers the energy is called as donor. The one which accepts the energy is called acceptor. But if they are at a distance of less than 10 nanometer, these proteins will start interacting. So we are doing an experiment like where you add a drop of drug. The moment you add the drug, the actual FRET starts. The FRET means the donation of the energy from the donor to the acceptor will start. But if you add a drop of drug, because of the vibration, your sample will go out of focus. If you are using this device called drift compensator, it will make sure that the sample is in focus throughout your experiment. Now, when you in this particular case, see, a drug is added, immediately the energy which is there in the cyan fluorescence protein or a CFP will transfer the energy to a protein which is called as yellow fluorescence protein or YFP. So the intensity of the cyan fluorescence protein, because it loses some amount of energy, will become lesser, whereas the one which gains the energy will become brighter, and you will be able to calculate how much of energy is transferred from a CFP protein to the YFP protein. This is called fluorescence resonant energy transfer, an experiment which is conducted to understand the vicinity of two proteins and whether these two proteins are really talking or not. Okay, and this is an example where you can see atherosclerosis. By giving a small light stimulation on the artery, you can stimulate the atherosclerosis. Then you can see the platelets are accumulating. Initially, they were moving fast, but now you are inducing atherosclerosis where the platelet will start accumulating, blocking the artery. So these kind of imaging, so where the microscope will no longer remain as an imaging device, now it can even be used to perform different experiment where you can use the light to induce different functions of the light. In this particular case, by inducing a specific wavelength in the 1s region, you can stimulate the atherosclerosis, thereby the platelets will start accumulating, aggregating, and clotting, and thereby it can block the arteries. So these kind of imaging can also be done with your confocal microscope. This is a cell division where you can see you can follow the cell division in 3 dimensions and you can record them. They were for live cell imaging. It is not necessary that like you always have to have fluorescence. This is a pure phase contrast image where no fluorescence is used. Neurons are growing over the depth and they are bifurcating, they are dividing. So this is a live cell imaging of neurons which is growing in a dish, provided you give enough nutrients to it. Now the question is, what next? What are the latest technologies that are available? A new technique is called spinning disk confocal microscope is available for fast imaging, where instead of allowing one point laser to fall, it can allow multiple points. A disk which rotates at a speed of 4000 RPM, it rotates. Now you can see proteins which are moving at a higher speed. Okay, so I'll just switch over to like show some of the images that are acquired through spinning disk confocal microscope. Spinning disk confocal microscope is basically a high speed. This is how it looks. A disk which rotates at 4000 RPM will be there. A camera which is used here is cooled to minus 110 degrees centigrade. These are called deep cooled cameras, and the collection capability of these cameras are 95%. That means every 100 photons coming out from the microscope, 95 photons are being collected, and they are cooled to minus 110 degree. It has an ability to go up to 2000 frames per second. So that means you can practically collect the dynamics of your cells. Look at this image. This was acquired with the live cell imaging, where the cell division is acquired with 2 different colors. You can see the entire cell division is monitored by a spin disc confocal microscope. Not only that, if there are some vesicles, if you are seeing some exocytosis, endocytosis, you can actually follow the vesicles and then you can see whether they are coming toward the membrane or going away from the membrane. They are there in the cytoplasm, whether they are in the plasma membrane, whether they are going and accumulate to the nuclear membrane. So there are experiments called photoactivation, photoconversion, where you can, yeah, any question? Okay. So you can also do some photoactivation, photoconversion experiment where you can specifically activate a specific region and then track where these molecules are traveling. So this is called a photoconversion, where a particular vesicle is, when you activate it, it will turn from green to red, and you can follow it. So in this case, initially it will be green. The moment you activate it is red, then you can see how these vesicles are dividing. This is a classical example of the C. elegans embryo, where the cells are going into meiosis. The first division and the second division can be clearly seen with the spinning disc confocal microscope. See here, initially it is one cell, divides, and then subdivides. Okay, that's how a spinning disc confocal microscope can work. But the question is, suppose if I have a thick tissue, nowadays people are working on different tissues, like it may be liver, it can be pancreas, it can be skin tissues, it can be brain tissue. So the conventional microscope has got a limitation of not being able to go deeper into the tissue. How do you address this one? This is addressed by a new technique called multi-photon microscope. Instead of using one photon, we use a multi-photon microscope. Why do we need this one? In conventional confocal microscope or the visible fluorescence microscope, we use a visible light. This visible light excites excellently at the surface. As you go deeper, it scatters more. The excitation efficiency will be lesser. Lesser fluorochromes are excited and you collect lesser signal. As you go deeper, your emission is lesser and you may not be able to see. That is why if you are using a conventional laser scanning confocal microscope, you can go only up to 150 to 200 micron deep. What about those tissues which has more than 100 micron? Suppose if I want to image the whole mouse brain. Nowadays, people are also trying to image the live mouse. It is called intravital imaging. In such cases, you have to have different techniques. So instead of using a conventional visible light, which are in the 400 to 700 nanometer, we'll be using infrared lights. This infrared light will go deeper into the tissue, collect the information. Now it has an ability to go up to several hundred millimeters. So in this case, if you see here, this is the brain tissue, the granule cells expressing membrane targeted EGFP in the dentate gyrus. So you are going deeper into the tissue, you will be able to construct the entire image. This is the brain tissue. This is the zebrafish head. The complete zebrafish head is imaged, and now you can have an access to go see the 3D image. So this is a DIC image, and this is the second harmonic. This is called label-free imaging. This is a muscle band of a Drosophila embryo. This is the Drosophila larvae, where you can see muscle bands and subbands. This is myosin and actin bands here. It can even be imaged in live. See here. So there are two signals of blood flow in the zebrafish embryo. Now I am imaging here. This is the blood which is flowing. Other one at the bottom is the embryo. So this is the muscle is the red one, and hemoglobin is the green one, where you can follow and you can track them. Here, this is the muscle and this is the hemoglobin that is. Nowadays, like tissues are so opaque and you may not be able to go deeper into tissue, a technique called clearing technique. So where you can treat these embryos, this is actually a mouse embryo. This is in PBS. And then when you treat them with a specific solution, clearing solution, it can become transparent without losing the morphology of the actual tissue. This will help us to go deeper into the tissue, and one can image now up to several millimeters. So in this case here, this is the cortex brain. You are imaging right from the top. here. And once you have, you can construct a 3D. That's how a whole brain imaging can be done. See now, this is the imaging the surface, and you are going deeper into the tissue, and you will be able to go up to 8 mm deep into the mouse tissue. So this is the cross-sectional view of the hippocampal brain part, and this is what you are conventionally observing 2-photon microscope. This is what you are observing with a transparent sample, and now with transparent, with proper treatment with a multi-photon microscope, you can go up to 8 mm, and you can image the entire structure of the mouse brain. So people are also performing some experiment called electrophysiology, where they will introduce some electrodes and then find out whether there is an action potential or not. It can be on a live tissue or cultured cells, where you want to see in which particular neuron the action potential is generated. So you can keep on recording this one by introducing a wavelength. So here I can stimulate a specific part of the brain and then see which part of the brain is responding. You can collect the electrical pulses. This can even be done on a live mouse. So can you see here? This is the microscope objective. This is a mouse head, and a cranium is completely shaved. And then you have an access to see the mouse brain, and put a drop of water, and then this is the water dipping object. The objective will come and touch the mouse brain, and you will be able to observe. You will observe to see the neurons when the mouse is live. And you can give stimulus. It can be an olfactory stimulus, or it can be physical stimulus, it can be chemostimulus. In all these cases, by giving a proper stimulus, and you can practically see which part of the brain is now responding. And you can also put an electrode here and then see what is the electrical pulses which is coming here. So this is called intravital imaging. And nowadays, like with the genetic engineering, you can actually generate action potential. You can silence a particular gene, and you can even activate a particular gene. So in this case, see here, what I can do is I use particular specific light. So in the absence of a light, the mouse, it is in completely action potential is not there, it is in silence. The moment you give a specific wavelength, the mouse starts responding. Okay, so that means you have a complete control. These are the two different proteins. See here, the moment you give blue light, the mouse will become completely active. The moment you give yellow light, it can be. So you can practically generate an action potential, or you can silence it. So this is what called optogenetic experiments. A protein called channel rhodopsin and halorhodopsins are being used to activate it a specific wavelength. Okay. So coming to the live cell imaging, the microscope can have a stage top CO2 incubator. The stage top CO2 incubator will maintain carbon dioxide concentration, temperature, and humidity, where you can culture the cells, and you can do multi-point imaging. And you can do a live cell imaging, as I told you. You can do perfusion experiment. And in those cases, like where you want to get a high resolution, super resolution microscope is also now into existence, where you can even get up to 120 nanometer resolution with super resolution. There are technique called SIM, there are technique called STORM, technique called STED, which can even resolve up to 10 to 15 nanometer in XY, and up to 200 to 300 nanometer in Z. See here, this is the conventional microscopy, this is the super resolution microscope. See here, this looks like only one dot in the conventional confocal microscope. When you switch over to super resolution, it is actually two dots. It can be resolved better. This is a pollen grain which is imaged. Okay, so other last function what microscope has, if you have a larger tissue, so if you have seen it in a smaller objective, it looks big, but you will not be able to see the smaller information which you actually see it in the higher magnification. In this case, you have a brain tissue with a low magnification object. You can see the whole brain, but you cannot see the cellular structure. But when you go for high resolution object, you should be able to see them. This is called macro to micro imaging, where you initially observe with the lowest magnification, image with the higher magnification, collect the high resolution image, seamlessly stitch it just like your Google map. Collect information and then stitch it from each point, and finally you form an image. That means you have the information of the whole brain tissue without compromising the resolution. If you keep on zooming it, digitally zoom, it will never pixelate, you will get the full information. This is an image of the mouse embryo. So 4 images were taken just to get the complete mouse embryo. Okay. And last part is I'll just show you like the latest development is artificial intelligence. Okay. I'll just move on to the next slide before I conclude. And then I'll show how artificial intelligence are being used in the field of microscopy. Any question till now?
No, sir.
Okay. So let me just move to. Can you see this slide by any way? I don't think you will be able to see. I think I'll just stop sharing and then. Okay, now I think you should be able to see it. Can you see it now?
Yes, sir.
Yes, yes, sir. Yes, sir. But not in slideshow, sir.
Yeah, now I see. Okay, I'll just tell you like why artificial, like you, I think all of you must be knowing what is artificial intelligence. And in this case, you have an image. This is a microscopic image. This is an image of a mitochondria. Like there are several cells are there. In each cell, you have multiple mitochondria there. The question is like I have an experiment where I want to see the effect of my drug onto the mitochondria. That means in a normal condition, I would expect the healthy mitochondria, and probably like because of some treatment, I would expect the number of mitochondria will reduce. The total volume of the mitochondria might reduce, or vice versa. Initially, like I have some particular disease, like where the mitochondria is reduced. Now I give some drug treatment, then gradually the number of mitochondria should increase, showing it to the cell is coming to a normal or healthy situation. That means you need to identify, you need to collect information from each and every mitochondria. So the conventional way of doing it is you just find out what is the intensity of these mitochondria, and I'll tell my computer to identify all those regions which are showing a specific intensity. For example, if you see this sample, the darkest region will be showing you an intensity as 0. The brightest region will be showing an intensity of 4000. Now I tell my computer, so considering some amount of noise between 0 to 100, show me all those regions which are showing an intensity between 100 to 4000. It automatically picks up all those regions and says, these are the regions which are showing an intensity between 0 to 100 to 4000. That's how this is called segmentation, or this is segmentation by intensity. But what happens in some regions, some signals are not being collected properly. So my question is whether can I develop an algorithm through artificial intelligence where I should be able to train that irrespective of its intensity, identify all those regions where mitochondria is there. That is my current demand. So with a conventional software, I cannot do it, but can I do a training to my software? That basically artificial intelligence is called machine learning, where you train a software and it will start thinking the way in which we have taught them. Okay, so now question is how do I train them? So I'll give you an example here. So this is our brain. So how do we identify a dog as a dog? How do we identify a cat as a cat? Or suppose if you are allowed to touch a small candle light, immediately you withdraw your hand. You will not even wait till the time your brain will tell you that like, okay, there is a fire, you don't go there. That means we have been trained, our brain is trained when we were small kids, so that we have a trained information to decide and immediately react. Now let's see here what happens. So there is, these are the pictures of different animals. It can be an animal which can be a cat or a dog. So this is image type. Now we have a learning phase. Assume that like my mother is training me, what is dog, what is cat. So then there is a ground truth data here. So what it is like, so our brains are being trained by looking at the image or by looking at the object, what is dog and what is cat. Now this is, now initially we will not have any cue which one is dog, which one is cat, or which one is dog. But after training, we will have a pre-trained neural network. So we call it as neural network. We have a pre-trained neural network which has an ability to distinguish what is dog, what is cat by looking at an image. Then how do you test it? So we challenge it. This is called inference phase. Now I put a small toy, then I will ask this neural network to tell what is this. What it does with this previous training, what it does is it will try to match and mismatch and then try to find out. And so this is my pre-trained neural network. Using this pre-trained neural network, it will try to apply the knowledge it has got on top of this one to see similarities and dissimilarities, and then say it is a dog. Then my question is, what is the surety that this is a dog? How can you show? What is the assurance? And I say, what is the probability? So then it says there is a high possibility that it is a dog because it matches more than 90% of the features of the dog for which I have been trained to identify it as a dog. Now I'll put another picture. So this picture, then again it will use the same, because now I have trained it both with cat as well as dog, but I have not trained it with the tiger. But it will again apply the pre-trained neural network onto this image and then try to find out whether there is any similarity between the dog and this animal. It doesn't know whether this is a dog or a cat. It will find lesser similarity with the dog. Then it say, but fewer similarity with cat. It says it is a cat. Then I question, what is the probability? It gives you a value, lower probability. It's to somewhat it resembles cat, but it is actually not a cat because it does not resemble. The probability it is not more than 50%. Okay. Now I'll put another small cartoon. Okay, can you tell me what is this one? The same trained neural network will apply the same logic onto this one and say this is a cat, but low probability because it does not resemble the real cat with which I have been trained my neural network. So this is called as artificial intelligence based neural network, where we train the neural network to identify the object, and then finally in the inference phase we'll check the probability, and if the probability is high, then I say the trained neural network is now giving us a reliable data with which I can actually use it for my routine scientific observation. Okay, any doubt in artificial intelligence? How does it work? Now the question is, how do we apply it onto the biology? Okay, now let us try this one. This is what I am doing. So now in this case, I have mitochondria. What I do is I will take around 10 images where I have imaged them with mitochondria, and I label all these regions which are mitochondria positive region, and then I say, see, these are the places where you can see it as yellow, they are mitochondria. Now initially it is untrained, it is unable to identify where the mitochondria, where the background is there, but I use around 10 images which is mitochondria positive, and I label all of them by using a proper algorithm, and then I say the yellow ones are the mitochondria, the dark regions are not the mitochondria. Now I have a ready neural network. Now I'll have to test it. What I'll do is I will use a different image other than the one which are used for training. Training, I have used a set of images that are positive signals. Now I use a different image which was not at all used for my training to challenge, to test my neural network. And I say, show me what is this image. Then it immediately apply this algorithm. It says wherever you have the red regions are the one which resembles mitochondria to the most. The violet regions are the one like which is partially mitochondria like. Blue are the one which are not at all mitochondria. So it gives you a precise heat map. This is called heat map. The red regions are the closest proximity to the reality, and the blue one is the closest proximity non-reality. That means these are the background, these are this one. So that's how artificial intelligence based techniques can now be used to identify the objects in a more precise way, in a faster way. Now what happens, once you train this and once you test it, now the neural network is a reliable neural network. Now you take tens and thousands of such images, you will be able to automatically analyze the total amount of mitochondria is there within a fraction of seconds. So there is one example. I don't think whether I have this image or not. So we have a kidney tissue. We have taken the kidney sections where you have labeled glomeruli. Now, so I want to treat them because maybe it's a mouse kidney. Now I have probably induced diabetic. Because of this diabetes, like the glomeruli is now has like reduced it has shrunken. Now I have taken another mouse where I have treated them. Then I want to see the number of glomeruli in the treated one compared to the actual untreated one. So the volume of the glomeruli, the number of glomeruli is my interest. Now what I do is I will take first a kidney tissue. I train my neural network. These are the places where my glomeruli are there. Once I train them, now I have hundreds of tissue sections. I will just acquire an image and then I'll put it into the artificial intelligence box and then say, use this trained neural network to identify the glomeruli and tell me how many glomeruli are there. Once I have this one, I can tell software what is the total area of the glomeruli of the treated ones, what is the total area of the glomeruli of the untreated ones. The total number of glomeruli on the treated ones, total number of glomeruli on the untreated ones. Like this, I will get an access. You get a precise information. You can avoid manual error, because suppose if one person is made to sit in front of the microscope and has to analyze 10,000 images, that maybe initial data are more accurate. As the time passes, the accuracy keeps on decreasing because of the fatigueness. One thing. Second thing is, if you assign 10 people, each people in there will be interpersonal differences. Each person can have a different way of counting, seeing it, or different perception to identify it as a positive or negative. There can be error. But now what you are doing is you have one trained neural network and you have validated it and you have found the probability map is more than 95%. That means out of 100 cells, 95 cells are being counted as glomeruli positive. That means I can rely this neural network. Henceforth, I will use this neural network for all future quantitative studies. Not only this one, suppose you have a collaborator who is working elsewhere in other countries. Then you say, so there should not be any difference between my understanding, the way in which I count the number of cells and the way in which they are concerned. I say, I have developed this neural network, and for all your experiment, if you're validating my experiment, please use this neural network so that there is no second variable. The variable is only a person to validate it. So that's how artificial intelligence are nowadays being used extensively in biological quantitative estimations. Okay, with this I conclude my presentation. To begin with, I'm sorry, like I just wanted to give an introduction on the microscopy because I was not sure like how much time I'll be able to show, because my main intention was to show the advances, but I didn't want you to get lost if I bring you directly to the fluorescence microscopy. I took around 5 minutes to explain what are the different kinds of microscope. So I started with bright field microscopy, and different morphology of microscope. It can be an inverted microscope, upright microscope, a stereo microscope. In case of bright field microscope, it can be a transmitted light microscope where I allow the light to pass through the specimen. I use a stained specimen and then collect it. That is called bright field. If I am not staining, then I use technique called dark field phase contrast or DIC, where I will use the oblique illumination and then collect the information of the unstained region. Then finally, if it is a reflected light, instead of allowing the light to pass through, I use a different light path where I introduce the light from the top and allow the light to hit the specimen, which can be opaque, and collect the reflection coming back. Finally, if I want to label specific different regions, I use fluorescence property. These are the chemical substances that has an ability to go and bind to different region of interest. And then I use this property and then made it to go in different bind region and then use a fluorescence microscope to observe them at different colors. If they do not have an affinity to go and bind, then I use the immunology method where I use the antibodies, tag the fluorochromes with an antibody, and then use a primary antibody to go and bind to a specific region of interest, and use the secondary antibody to go and bind onto the primary antibody, and then see the colorful regions which has got a specificity to go and bind to different region of interest. Once this is done, then I use it for fixed specimen. But if I want to use it for live specimen, I use the central dogma of molecular biology, where I used proteins to express, and I had a gene that has got an ability to express a specific fluorescence protein. I have co-expressed by fusion protein module, where I have co-expressed this protein of my interest along with the fluorescence protein to see. This has opened up a new type of imaging called live cell imaging, where you can actually see the fluorescence which are in live condition. I should be able to image it in live condition. So I used a different kind of microscope called live cell microscope. What we do is we had to maintain the integrity of the cells. We could have a stage top CO2 incubator where the carbon dioxide concentration, humidity, and temperature is maintained. You have an incubator on top of a microscope. You keep on observing while you are imaging. This also has given us an option called perfusion experiment, where you can use drug delivery experiment, where while you are imaging, you can introduce a drug and then you see the efficacy of the drug on a live cells to see whether they're affecting or not. Not only this one, if at all you are treating them with like natural killer cells, you can introduce the natural killer cells, label the cancer cells with an antibody which has got a specific color. The natural killer cells has got an affinity to go and identify the antibody bound cancer cells and kill it. The moment it kills, the blue color will become red, showing that the targeted cell death is now achieved. Once this is done without whatever we are doing on a regular microscopy, can we achieve a better resolution? Super resolution microscopy was introduced. Super resolution microscope has helped us to achieve a better resolution down to 10 to 15 nanometer. That means you can practically some proteins. Protein interaction can have been seen. So once this is done, then what we thought is how to do the analysis. No doubt you must have done an experiment, excellent experiment. You have documented it with a very good microscope. Your images are very good. Your experiment has done, but there could be an error while you are imaging. There could be an error while you are analyzing. So in order to avoid this human error, people have started using artificial intelligence as a tool. Artificial intelligence uses basically a neural network. Initially, it can be a neural network which is completely untrained. And we train the neural network to identify a specific object. Once you train them, we have to first go for inference phase to validate it, whether the training neural network is proper or not, by challenging it with an unknown specimen and see whether it identifies as a specimen of our interest. If it gives you, then we find out what is the probability map. And if the probability map is more than 75%, I consider this as a reliable data and reliable neural network. Later on, I will use it for studying hundreds of such images for analysis purpose. So that's how an artificial intelligence based image analysis has become picking up and becoming popular nowadays. So with this, I conclude my presentation.